Nanobiology. My name is Joris Pothoff. I'm the Nanobiology Bachelor of Science Director, and the other hosts are Joanna Colgrove, the Nanobiology Program Director, Stan de Schrijver, a Nanobiology student, and Gijsje Koendering, a Nanobiology lecturer. One practical item, if you have questions, you can type them in the chat, and we will answer them live, and in some cases also in the chat.
Joris, je staat op mute.
I saw it, sorry. I will go back. Sorry, I was muted, classic mistake. Welcome to the presentation of Nanobiology. So, I'm Joris Pothoff, I'm the Nanobiology Bachelor of Science Director, and the other hosts of today are Joanna Colgrove, Nanobiology Program Coordinator, Stan de Schrijver, Nanobiology student, and Gijsje Koendering, a Nanobiology lecturer. So if you have questions, please type them in the Q&A, and we will answer them live in the Q&A session or in the chat. So, nanobiology. What is nanobiology? So, in a popular way, we say it is using math and physics to understand biology. And first, I would like you to bring into our world. So, how small is nano? Well, we have our Earth in distances in Earth we measure in kilometers. Then we have the living organisms on Earth, for example, ourselves. We measure that in meters or centimeters and millimeters. But then we actually have to go smaller. So if we then zoom in and look at 1 square millimeter, then we can fit about 1,000 human cells on 1 square millimeter, or about 500,000 bacterial cells, or 100 million Sarskoff-2 virus particles. The reason that we are now doing this online and not that we can greet you live in a lecture hall. But if we then, we already approaching nano. For example, the SARS-CoV-2 virus particle is about 100 nanometers in diameter. But let's go even smaller. So then, for example, per human cell, there are 46 chromosomes, 23 from your father, 23 from your mother, and together that is 2 meters of DNA. These encode 22,000 genes, and these produce together 400,000 messenger RNAs, and those produce together about 42 million proteins, and that's all in one cell. And we're not even talking about the lipids and sugars and other macromolecules inside that cell. So if we look even further, for example, in a red blood cell, there are proteins, the hemoglobin proteins. They form these quaternary structures, so they 4 proteins bind together, as indicated here in the right corner. And these are the proteins that bind, they have an iron inside, which binds the oxygen and make sure there is oxygen transport. And here you already see that we're looking at nano actually also at the atomic level. So how do we work and think? Well, it all started, of course, many of the biology with microscopy, light microscopy, so what can we see? But we go even smaller. And for that, I would like to give you a timely example of the SARS-CoV-2 virus. So here in the red, we have the SARS-CoV-2 virus with these spike proteins, and we know that it binds the human ACE2 receptor. And that is the way of infection, but it is not a permanent binding. So it binds and it dissociates, it binds and it dissociates. because the binding is due to hydrogen bridges. So basically we could show that in a way as this in the bottom, so binding dissociation. So how is it then able to infect a cell? Well, if we look, in some cases when it binds, we get this inclusion. So then the virus particle is taken up and then is released inside the cell, and then we have an infected cell. But how does this really work? Well, then we really have to zoom in a spike protein of the virus and the receptor. And if we then do that, we can visualize how that looks like. So here in red, we have the human ACE2 receptor, a part of it. And here in gray, we have the ACE2, we have the spike protein of the virus. And if we then really zoom in, we can see here how these two interact. And we see the amino acids and the hydrogen bridges, how these two connect. And then we can measure how strong is that connection. So basically what we then measure is with what strength, so we would call that affinity, does it bind, and how does it dissociate? So think of it as two magnets. We all have played with magnets. They're together, and then you have to also have a force to pull it out of each other. And this is basically what this graph does. So if we go the line upwards to the peak, that is the binding strength. And then the line downwards is the dissociation. And this is of the wild type SARS-CoV-2 virus, or the normal variant, as we call it. And then what has been done here, that we have these human ACE2 receptor on the surface, and then with increasing concentrations, each of these lines is an increasing concentration of this spike protein. And you see that that we get a certain binding and strength and dissociation curve. So what happens if we put there one mutation? So we mutated the asparagine at 501, so at amino acid 501, into a tyrosine. So this is not just a randomly chosen mutation, but this is what we in the popular news is called the South African variant. And the South African variant, as most of you have heard in the news, is more contagious, is more infectious. So basically, if we then look really at the molecular level, what's happening, actually at the atomic level, we see that this interaction in the wild type, in picture B, image B, is different. So when we have to look at picture C on the left, we see there's a new interaction between two amino acids, and also these two aromatic rings, they are stacked onto each other. So this gives a different type of binding. And if we then go back, if we then go to the right, to the new affinity and dissociation figure, we see basically the same way of binding strength. So here it's about, the peak is at 25, here it's even a little bit higher. So it binds with the same strength, but the dissociation is way slower. So that means if we would capture that in an image, that it binds with the same strength, but it stays bound to the human ACE2 receptor much longer. So, and how do we then differ also from other studies is we can now also capture these kind of data into mathematical models. And with these models, we can predict how biology will react in other cases if we make other mutations, etc. So basically, we use the language of math and the concepts of physics to understand the complexity of biology. And for that, we really need a new type of scientist or engineer. And that is basically what nanobiology is all about. So other items or other studies where nanobiology matters is, for example, in medical imaging. For example, the MRIs, we make there's brain scans, and with all these complex images, we can do machine learning, and which is a form of artificial intelligence to really understand how such a complex organ as the brain is changing in these images. Or bio-inspired engineering, but also evolution as a design tool. So train phages, and phages are viruses that kill bacteria, and you could train them to kill that bacteria that are pathogens. Or in the very big genomic data sets that we have, and genomic data means many people have their genomes now sequenced, or we know exactly what the tiny differences in the genome between different people are. And can we use that information to see what are genes that cause a disease, or that help us to make drug choices, etc. So these are all items where nanobiology really matters. And therefore, the TU Delft and Erasmus University Medical Center have combined their strength. The TU Delft, with expertises in nanoscience, biophysics, technology, and Erasmus University Medical Center, the main expertises are biology and biomedicine. So you get the best of both worlds. One question we often get is, well, 2 locations, we're a joint degree, so how is that practically working? Well, you have about 50% of the classes are at the Erasmus Medical Center, the other 50% at the TU Delft. It's here in this map, the distances, but it's only 1 location per day, so it's not that you then during your afternoon lunch have to travel to the other location. Actually, the TU Delft and Erasmus Medical Center are quite close to from each other. So from door to door, and then I mean from your door if you live either in Delft or in Rotterdam as a student, to the other university is about 30 minutes by public transport. And if you want to bike it, it's about 50 minutes. So this is these distances are quite small, so it is easy to reach. The courses, a little bit about our program. All courses are in English. So we are an international program. That means that there are Dutch students, but also international students. But also the teachers, we have Dutch teachers, we have international teachers. And there's a strong integration between subjects. And we like active learning. So have problem classes in which you solve together with a group of students, fellow students, and teachers and teaching assistants problems. So in year 1, this is a little bit the division of the courses you will take, and then we divided them based on a topic. About a quarter is biology and math, also a quarter. Physics is in year 1 a little bit less. And then what we have, what we call bridge courses. And bridge courses is where at least 2 topics of biology, physics, and math are integrated. For example, the physical biology of the cell. So you can imagine that we have biology and physics that is integrated, and often you need math to solve the equations there. Then in year 2, more than a quarter is physics. We have math, biology, and additional courses that are also bridge courses that connect all these topics together. And then with year 1 and year 2, you have a very strong foundation already in nanobiology, in the theory of nanobiology. And then in year 3, it's basically 3 items. One is a minor of 30 ECTS. And this is a minor is a half a year of courses, of projects that you can choose yourself. So there are many minors, and students are free to choose their own minor. Then there are elective courses worth of 10 ECTS, and these are courses that you can choose based on your own interests. So if you like to do more biology, then students choose more biology courses. If physics is really your thing, you choose physics courses. And the 3rd year ends with performing your Bachelor of Science thesis, worth of 20 study points. So the program goal is to prepare students to take part at an academic level in the fields of bio-nanoscience, biophysics, and biomedicine. Mathematics. So what will you learn in the mathematics courses? Calculus, linear algebra, differential equations, statistics. In physics, you will learn about mechanics, optics, thermodynamics, quantum mechanics, but also chemistry. We have some chemistry courses where you learn all about atomic structure, organic reactions, biochemistry, and of course, biology, starting from molecular biology, genetics, evolution, development, and this together gives you a solid foundation in nanobiology. So there are several skills you will learn. So it's not only the topics per se, but also presenting, programming, writing, practicals. And here you see some courses from year 1, 2, and 3, and then with a color code, what type of skills you will learn during these courses. And we often get a question like, programming, what level should I have before starting nanobiology? There's a very simple answer. We will teach you how to do programming. So just your high school level of programming, which for most students is 0 experience, then you enter, and we will of course teach you how to do programming. And the 3rd year of the nanobiology ends with a bachelor end project and writing your thesis. For a bachelor end project, you join a nanobiology experimental or theory group for 20 weeks. So note, all your teachers are also scientists and have their research groups, and in which they study their favorite subject. And so basically, you're looking through all the list of teachers and what they are studying in their research groups. And then as a student, you choose a topic you like and join that group for 20 weeks to do an independent research project together with a supervisor, such as a PhD student or a postdoc. And this is either in Rotterdam or in Delft. And based on that original research, you write a thesis which you have to defend, which is always very fun because also parents can be present and friends, and they can really see what you have learned in all those years. So we have multiple teaching methods. In class, there's about 22 hours per week. These are lectures and also learning by doing, problem classes. So you're sitting in small groups and the teacher provides problems that you have in math, physics, biology that you have to solve together. And of course, our teachers and teaching assistants to help you out if you're stuck. Self-study, we estimate is about 18 hours a week, maybe a little bit more. I think Stan can tell you more about it. He will show you how his average week looks like. And we have practical work in year 1. It is wet lab and computer modeling. So wet lab really means with wide laboratory code, you go into the laboratory pipetting. In year 2, it's computer modeling, instrumentation, and microscopy. And then in year 3, of course, your bachelor end project, in which you're really in a lab or in a digital lab, if you want to do, for example, mathematical modeling. We also have an honors program for excellent students who are interested in doing a little bit more. So how you're assessed? Well, with exams, of course. We have an octal system, which means the year is divided in 8 parts. And that means then that there are exams every 5 to 10 weeks. So some courses are octal courses, so they're only there for 5 weeks. The overt majority of courses are actually quarter courses or semester courses, so they're for a whole quarter, so that means 10 weeks. And exams are, for example, open questions, multiple choice, but several courses also have assignments as part of the assessment. in which you have to write something, present a project, either in groups or as an individual, and the grade you get for that assignment also counts towards the grade of your for the whole course. So what after finishing your bachelor? Most students, if not all, continue doing a master. Many are currently doing the Master of Nanobiology, again at TU Delft and the Erasmus Medical Center. But other options are Erasmus Medical Center Research Masters, Molecular Medicine, Neuroscience, Infection and Immunity, or TU Delft Masters, Applied Physics. You have to take some extra courses, so some students see like, I really like physics much better. And then in their third year, they take a minor in physics, do elective courses in physics and a bachelor end project in physics, and then they can continue to the applied physics master, but also biomedical engineering and many, many other national and international Master of Science programs. Job opportunities. With a Bachelor of Science, graduates will enter high or nanotech industries, sales, education, but actually everybody continues with a master. And then there are many more options, for example, biomedical and nanotech research and development at companies, access to national and international PhD graduate schools, so becoming a doctor of philosophy in science, government jobs at Dutch research organizations, consulting, finance, education, and if you decide to do, after your Master of Science, a PhD, then you're becoming a full scientist, and most of these pursue an academic research career or a research career in the private sector, such as biotech companies, pharmaceutical research, pharmaceutical companies, etc. We have not so much data because what everybody is doing, or we have not so much graduates because the first Master of Science graduates were in 2017. But here are some examples in which our graduates ended up. In research and development in companies, for example, at Eurofins or Lumix, many, about at least 50%, continued doing their PhD and are now working in labs in the Netherlands, Denmark, USA, Switzerland. I also have PhD students, which are nanobiology graduates in my lab. And I think most lecturers have government, consulting and finance, for example, Ernst & Young Financial Services, and then people think, why do you end up there? But think of the general skills you get when you finish your nanobiology. You have analytical, abstract and conceptual thinking skills. You're not afraid of data processing and can handle large data sets. You can do modeling and programming, presenting and writing. So these are all highly valuable skills for various corporate and public positions. So therefore, nanobiology students or graduates are really wanted by both universities, but also in private companies. So what makes a good nanobiologist? Well, you could have guessed a love for math, physics and biology. For Dutch students, you need the following VWO profiles, NG in Dutch Natuur en Gezondheid, plus with Physics and Mathematics B, or NT in Dutch Natuur en Techniek, with additional subject Biology. The advice is, we always say a grade of 7 or higher makes that your study success in nanobiology will be fine. And yes, that we have also cases in which that's not sufficient, and with sixes that students really brilliantly go through the study, but we see a correlation between your grades in fifth class, and at the end of fifth class VWO, and the study success in nanobiology. And a pioneering spirit. We are a very young study, and we are a very unique concept in which we combine math, physics, and biology. And that is unique in the Netherlands, Europe, and even the world. And the pioneering spirit is also to help improve education with us. For example, after every course, students will get a questionnaire to also grade the course, so we can improve the course next year. And that is what we think is really valuable, this interaction between teacher and the program and the student. And of course, an interdisciplinary mindset, because you have to integrate all these topics into one package. Some relevant facts. We're a focused small-scale study. We have 120 students in the first year. We're an international bachelor, so means there's a combination of Dutch and international students, and our main language is English. And then we often get the question like, oh, but is it then so, students are a little bit afraid in the beginning, like, yeah, but then I have to speak English, and is that not difficult, etc. Well, very simple. You had English in your high school. If you graduate with a 6 or higher, you will have no problems changing from Dutch to English. And we teachers will help you with that as well. If you don't know the words, etc., we help out, of course. We're a new and unique program that started in 2012, and accessibility for disabled students. And there's also binding study advice, which means you have to get 45 out of 60 credits in the first year. 120 students we can admit per year, and therefore we have a numerous fixes or enrollment cap. And we select them based on ranking. So how's that working? We have multiple criteria to determine the rank. One is an online questionnaire in which you have to answer several questions that include what are your grades at your final at 5 VWO high school. The other is a selection exam, often early March. And that's an exam in which you have to make you have to answer questions and solve problems, nanobiology problems, and for which we will provide you with study material. And also there it's based on a rank, so it's not that we say you need a 6 to be eligible for our course for nanobiology. We basically say we rank all the students from 1 till 200 something. Based on how good they did the exam and also the questionnaire. And then we combine these ranks, and we have a final ranking from 1 till this year 220. And this would determine whether you have a position next year as a student. Thanks for joining us today. So now Stan de Schrijver, a nanobiology student, will talk about his perspective of the study. Afterwards, we have a live Q&A, which is also led by Stan. After the Q&A, Gijsje Koenderink, a professor at TU Delft, will give a mini lecture. And afterwards, we have a live Q&A. And in the meantime, we also, when Gijsje gives her presentation, we will also continue answering questions. So thanks for joining us today. And then Stan could share his presentation and continue.
Thank you, Joris. Okay. Yeah. So I will talk about the student experience. While I'm talking, please feel free to send in any questions. We have more than enough time to answer any of them. I've noticed that a bunch have already come in. Great. So after my presentation, we will have a short time to discuss these. And then during the guest lecture at the end, we will still be able to discuss the questions. However, that will only be done via text. We will make sure that everything is published so that everyone can still read through everything. So yeah, okay. Hi everyone, I'm Stan. I'm a second year student in a bachelor's nanobiology, and today I would like to talk about why you would choose nanobiology, something about my own study choice, about my typical week, and something about SV and Hoek. So why would you choose nanobiology? Well, the word multidisciplinary has fallen a bunch of times now, but this is really something to keep in mind, because rather than studying one principle, you study multiple. So instead of just physics, or just biology, or just chemistry, or just mathematics, it's a combination of everything. So which is unique, if you will. We're also an English program, which makes it we're international as well. So in my year, there's students from New Zealand, there's a student from America, from Italy, well, from all over the place, which makes for a very international atmosphere, and so a very fun group of students. Also, professors are international. We have professors from Italy as well, from Sweden, etc. And so you get to know people from all over the world, and you are in this group of people with very different cultures and stuff, which is very great to hang out with. Lastly, the male-female ratio is about 50-50, which is also noteworthy here in Delft. So about my own choice of nanobiology, I knew that I wanted to do something within the field of medical sciences and technical sciences, and a combination between the two. I will get back to that in a second. But one of the breakthroughs for me, and so what I based my final decision on, was the fact that the program is so internationally oriented. So what I really value about the program is that you can keep your options open concerning where you end up eventually, in the sense that it is an international bachelor's, and so language, the level of English will not be a barrier anymore for any other types of education or jobs that I might have in the future. Yeah, and it's a very exciting field. So it's a new field. It's a field that is very much in rapid development. And so you read a lot about the field even outside of the studies. So for example, what we see here on the right is a protein that is called CRISPR-Cas, which is basically a new technology that can easily cut and paste DNA. So a few years ago, the finding of this enzyme was a very big breakthrough in molecular genetics, because this allows scientists to very easily change DNA. So we can easily reshuffle the letters of the genomic sequence, if you will, which was a really big breakthrough in science. And so this is an enzyme that we also study a little bit in nanobiology. We discuss it in a first year course, which is called genetics. So for me, that was last year. And then in 1 year, I saw a lot of headlines and a lot of articles about CRISPR-Cas molecules, and about new research showing that CRISPR-Cas is very efficient. And so this system specifically is very much in development. So you learn about something, and then for another year, you still hear and see a lot about the same protein. So that's what I'm trying to say about the field being very much in development, which is incredibly exciting. Again, interdisciplinary. You merge multiple disciplines into one, which makes that with broad interests you can very much enjoy the program. So like I briefly said, the fact that it's internationally oriented was for me the eventual gave me the eventual final decision of doing this program. But I wanted to do something in biology slash medical sciences slash something technical because of my first lab related experience. So in Dutch high school, you end your high school career with a PWS, as it's called in Dutch, which is basically your project, your final project, where you spend roughly 40 hours on one project related to one specific course. For me, that was biology, and I chose to do my PWS on CRISPR-Cas, which is again the enzyme that we see on the right, and my partner and I started to email a bunch of people and a bunch of institutes of which we knew that they were working with CRISPR-Cas. And eventually one university, the Radboud University in Nijmegen, they sent us back an email saying, hey, why don't you come and join us for an experiment? You can have a look around in the lab, have a look at what it's like. And so we went to this big university, being two 17-year-olds, and we came into this giant laboratory with electron microscopes everywhere and with big machines, which are very exciting. However, I didn't know a lot about it. So eventually we did look at cells and we did look at DNA and what CRISPR-Cas did, but I didn't really understand how the images were formed, how the computers worked, how the machines worked, for that matter, which really gave me the interest in the overlap between the biology field and the technical field. So coming back to the interdisciplinary, to the fact that the program is interdisciplinary, here at the bottom we see a quote from a student that's a year older than me. To complete a tough calculation is one thing, but to actually understand its biological significance is something else. In my experience, nanobiology teaches you exactly that, which is I think a very good description of what nanobiology does. We get tough calculations, but instead of just making them and not entirely knowing what they're about, we know what they represent and we know what these calculations and equations do and stand for, which is incredibly exciting, I must say. So my typical week looks a little bit like this. I have to admit this is based on the curriculum at the first semester of this year, so that run from September until January. We see a lot of blue and a lot of green. The blue consists of lectures and of seminars, where seminar is just a fancy word for working class. So what happens a lot is that we have lectures, afterwards we have seminars. So for example, if we have a mathematics lecture in the morning, we can have a mathematics seminar of the same lecture in the afternoon, where you get exercises and you work with these exercises, and professors and assistants walk around, and you can chat with them. You can ask them questions about the material. You can ask them questions about exams, etc. Then the yellow box is an on-campus practical. So in the first year, you have some practical work in the second semester, and then in the second year, in the first semester, you also have some practical work by a course which is called Electronic Instrumentation, where even in these times, we were allowed to be in the lab once every 2 weeks for 4 hours. So each 2 weeks, we had a 4-hour session in the lab, where we still did practical work. So even in these times, you still do practical work. And you see a lot of self-study. So the advice of the university is to spend around 40 to 45 hours a week on the program, which is similar to a full-time job, right? I think I spend a little bit more than that on the program, but it's only an average. Yeah, and then besides, of course, you might want to be socially active as well. because there is a lot more than just study. There is a lot more to Delft or Rotterdam than just studying. So what you see on the Tuesday night is that I have a committee night, which is part of Hoek. So SVNB Hoek is a study association from the program Nanobiology. So in Delft, we have a lot of associations. We have sports associations. We have music associations, etc. But there are also study associations. So each program has its own association. So physics has its own, nanobiology has SVNB Hoek. And this association organizes a lot of things. So we're very active, normally speaking. Now we are also very active, but then online, of course, like everything is. So the pictures that you see here are taken from last year and not this year. So what we have is a gala. We have a annual holiday trip. So last year, February, we went to Gran Canaria. We have parties. We have small activities like hitchhiking to Lille, which happened in last year March, I think, about a week before the lockdown was introduced. And all these activities need to be organized by someone, which is done by committees. So there's a committee that regulates the holiday to the annual holiday. There's a committee that organizes these parties. I myself was in a committee that took pictures at the Hoek events. So at parties, I would be there with my camera and I would take pictures of everyone. Currently, I am part of the committee that does the booklet of SVNB Hoek each year. There are 3 editions of the booklet where we interview people, where we write about interesting articles about scientific breakthroughs, etc. So in these committees, you work on something with a group of people, and so you get a bit of working experience, if you will, and a lot of collaboration experience, and you get to know fun people. So that's a win-win. The fun thing about Hoek is that we're a small study, so everyone knows each other very well. A lot of people are active. Again, an international environment. Plus, in Hoek, you can just talk to people that are older than you or younger than you. So everything that you wonder about, people have already lived through. So me as a second-year student can ask third-year or fourth-year students about future perspectives, bachelor and project experiences, etc., etc. So that's a very fun atmosphere. Here are 2 QR codes. One goes to the study choice page, where there's some information about whatever you need to know. There's also a link to a platform called Unibuddy, of which I am part, where you can just chat with me for any questions that you might have. I will share my email address later this session to do the exact same thing. And on the right, we have a QR code that leads you through our Instagram page, where we have some updates about scientific breakthroughs every once in a while, about open days, and about a lot of other education or nanobiology related things. So that was my presentation. Thank you for your attention. We will now move into the Q&A session. So for any questions that you might have, please feel free to share. I will go over the majority of questions myself first. But other people that are in this session, so the ones that are introduced by Joris Pothoff, will also be able to take questions. So you will probably see every one of us around. Okay, the first question, the difference between the program Molecular Life Sciences and Nanobiology. So Molecular Life Sciences is a program taught at both Wageningen University and Nijmegen. The difference between Molecular Life Sciences and Nanobiology is probably that Molecular Life Sciences has a lot more chemistry in it, whereas we are a lot more technically oriented. So we have a lot more, math, a lot more physics, and in that sense, we are a lot more broad and fundamental, given the technical aspects to it, if you will. Question number 2. Will you study at TU as well as Erasmus if you apply at Erasmus? Yes. So people apply. I see that Joanna wants to answer this question.
You apply to the nanobiology program through TU Delft. There isn't an application process through Erasmus. So there's in StudyLink, there's a piece, but the whole application is at TU Delft. TU Delft is the penning forwarder of the program. So that is where all of the sort of administrative side happens. You don't choose which one you apply at.
Great, thank you, Joanna. Which language do you program in? So there are multiple languages, the major one of which will be Python, but you will also get introduced to Java, to R, to ImageJ, which is a software that's used for microscopes a lot. So there are multiple, but the major one will be Python. Is it possible to join for masters from other faculties with his bachelor? Yes, it is. Joris already showed a bunch of masters that you are eligible for. But in the third year, there's a lot of freedom for you. You can also choose to take some courses which are needed for other specific masters. So you can change direction, if you will, and you can apply for other masters. However, for some programs, it takes you, you might need additional courses for some programs. Is there a numerous fixes? Yes, there is. This was already answered by Joris, but we let in 120 people based on a ranking from a selection procedure. This is a very specific question, whether VWO certificates would be enough to apply, or whether a word that I don't know is necessary. Joanna, could you take this question?
I can. Here. Yes, you can just use the VWO certificates. If your HABEO doesn't include the necessary coursework.
Well, that was easily answered. Thank you, Joanna. Will all of today's presentations be recorded and accessible after? Yes, they will be. This session will be recorded and will also be put on the website of the open days of the university. So you can just rewatch specific parts of it if you would like to. Is it still possible to register for next year? If yes, what is the limit date? No, it is not possible to register for coming year. I think that the last day that you can register for the program is January 15th of each year before you start in September. So for this year, you cannot register anymore. Are there any ethics classes, and how do you address the ethics of nanobiology in general? Yes, there are ethics classes, or yes, there is an ethics class in the second year. The course is called Philosophy and Ethics, where you indeed talk about, the ethics of, for example, trials on animals and, science in general. So ethics is discussed. Would anyone like to add anything to this? I see that Joris would like to say something.
Yeah, so we have in the second year, we have ethics and philosophy, which is the philosophy of science, ethics of science. But also already in the first year, we start, for example, already in the very first course, Introduction to Nanobiology, for example, what is plagiarism, what is fraud, and how to prevent that in your own work. So we really try to train nanobiology students in in very ethical human beings, that you that scientific and academic integrity is really we keep a very high standard there. So in various courses you will see this back. There's also in courses how to recognize frauds in scientific data. So so it comes back at various occasions.
Thank you, Joris. Next question, whether we are aware of people who did Mathematics A in high school and got a Mathematics B diploma via summer school. I don't know anything about this. Maybe someone can help me out here. I see that Joris would like to say something.
Yeah, so basically you need Mathematics B. So if you have a VWO diploma and then the certificate for Mathematics B, then it's fine. So you need to obtain that certificate. Oh, Joanna also would like to say something here.
There are also recently 2 new pre-university MOOCs, open source courses in the summer, one on calculus and one on physics, that we sort of strongly recommend to students that they take it, because it really goes from how you learn math and physics in high school versus how you learn math and physics in university, which is very different. So those should help everybody get to the same level at the start of the year.
Great, thank you. A question about the ratio of practical and theory. Joris, I think you will be able to give a much more useful answer than I would.
Yeah, so there are 2 practical courses in the first year, which are laboratory course 1 and 2. That is where you really are in a lab with pipettes. That is, in total, these are then 6 mornings or 6 afternoons. So, and then we call 5 hours of work, a morning or an afternoon. That is the real practical, but we also have computer practicals. And actually doing all these work classes and where you do problem solving is also a part of a practical, right? Because you first get the theory in a lecture, and then you have to apply the theory to a scientific problem. It could be biology, could be math, could be physics, and you have to solve that. So I consider that also as a practical, as you're busy trying to train yourself in solving problems. The ratio between lectures and work classes, problem solving classes, differs really per course. In some courses 50-50, others it is 70% theory, others is 30% then work classes. But the laboratory courses, these are for 80% practicals and 20% theory about practicals. So I think they have a little bit an idea of how you're busy at university.
Thank you. Okay, so while we will still continue for another roughly 5 minutes to answer these questions, you can still send them in. Like I said previously, in a little bit, we will have a guest lecture by Gijsje Koendering, while we will still be able to answer the questions via text. And if there's time left after the guest lecture, we will have another Q&A session like this. Next question is about the differences between biomedical engineering and nanobiology. We had this question yesterday as well. Joanna, would you care to weigh in?
Sure, happy to. I would say that biomedical engineering is more solution-based. So they're looking for a solution to a particular problem, and nanobiology is more fundamental of understanding how the cells work. And yes, you look for problems, and people do medical research as parts of their BEPS, but it is really at the sort of very fundamental molecular level.
That makes sense. Thank you, Joanna. Next question is about the average age of the first year international students. Maybe someone from the program knows this in actual numbers. However, in my experience, I don't see any difference in average age. But again, maybe someone else knows the actual numbers. Okay, I see that Joanna would like to say something.
I don't actually know that I have a better answer than you, Sten. I think they are generally sort of directly out of high school as well. I know we have a few students who come after having done some other bachelor program and then join our program. So there are some students who start a little bit older, but not much.
That makes sense. Thank you. Question about whether most people live in Delft or in Rotterdam. The most people live in Delft. However, some people do live in Rotterdam. Yeah, Joanna would like to add something to me.
I think they also live in Leiden, Den Haag, and various other places as well, sort of based on a whole range of things. So, you know, we're sort of right in the middle of everything.
Yeah, we are. That's right. One thing I would still like to add to that though is that so a lot of like I introduced, a lot of people are very active in the study association Hoek, and Hoek is majorly based in Delft. So most of the activities are around here, which makes that most people from the program also live around Delft. Also, because a lot of the nanobiology students join any associations, both in Rotterdam and Delft, but then again, mostly in Delft. The question about the differences between the program of clinical technology and about nanobiology. Well, clinical technology is similar. As it's also an interdisciplinary course, but the skill that we look at is fundamentally different. So for us, the cell is a central unit. However, for clinical technology, it will be the patient, and so a medical problem. But then again, the combination of mathematics with physics with biology is similar. It's just that the skill that we look at is fundamentally different from each other. You always like to say something.
Another difference is we train students to become scientists, so that is our primary goal. And in clinical technology, you will be trained to work in a hospital and applying medical technology and a new principle and physics and these things. You're allowed to work with patients if you finish your master clinical technology. So that is a fundamental difference in where you end up after graduating your study.
Okay, thank you, Joris. Given that it's 10 o'clock, I would like to give the word to Gijsje Koenderink, who will give a guest lecture. Like I already stated, please, for any questions, just send a text and we will be able to answer them via text as well.
Thank you, Stan. Can everyone see my slides?
Yes, we can.
So I would like to give you an example of nanobiology science. You've seen already a few examples sprinkled in the presentations of Joris and Sten. And I would like to take some time to go a little bit more in depth with an example that is particularly close to my own heart, which is material properties of living matter. So I'm a nanobiology lecturer and I'm also a scientist. I'm working at TU Delft in the bio-nanoscience department. So we actually work inside the building that you see in everyone's background. And I work there together with 15 other research groups, and all together, our purpose is to study life at the nanoscale. So my own research, you could say, is really centered on the question, what makes living matter? So basically us and cells that we are made of, what makes living matter different from non-living materials? What is the magic that makes living matter different? And of course, living matter is different in many different ways. But when we think about it from a physical point of view, from a materials point of view, we can notice a few unique features. So one of them is that our body has very special material properties. So you can actually test that, for instance, by pulling on your own earlobe. When you pull on your own earlobe, you will find that it is actually really soft. But if you pull very hard, it's also very strong. The harder you pull, the more it resists the pulling. And this is what makes our tissues somehow very pliable, but also very strong. And if we do incur some damage, such as a broken bone, which happened to one of my sons quite recently, fortunately, our tissues have the capability to heal themselves and repair themselves. And maybe even more amazingly, think about the fact that all of us emerged from a single cell that got fertilized, and then somehow this grew out into an entire human being. So living matter is soft, strong, self-healing, and self-growing. So where lies the magic that fuels all of this? And of course, the magic occurs at the level of the smallest living building blocks of life, which are our cells. Which, one of the first scientists who identified those was actually a scientist in Delft, and you could say he was a nanobiologist avant la lettre. This was Antony van Leeuwenhoek, and he was actually not a professional scientist, but a businessman. And in the evening and weekend hours, he would build little microscopes and look basically at everything that he could get his hands on. And he identified cells as the smallest living building blocks. Now, of course, nowadays, we have much more powerful microscopes to look at cells and look at the nanoscale building blocks that are inside them. So, for instance, on the left here, you see a microscope in our own lab, which is an optical microscope, where we usually use fluorescence microscopy to look at cells. And an even more powerful microscope that we also use in our department is an electron microscope, which can magnify up to 1 million times. So what do we see when we look at cells and tissues through these microscopes? Well, it depends actually on which molecules you stain, but the molecules that we stain with fluorescent dyes are actually proteins that form the mechanical frameworks of cells. So in this image, you see in blue 2 cells, with a blue label, and around them you see this yellow stuff, and this is what we call the matrix. So what you see here are actually proteins that form filaments. So outside in this extracellular matrix, we find these rope-like fibers that are made of collagen, which is a protein you may have heard of. And inside the cell, we have very thin filaments, which are called cytoskeletal filaments. And you can imagine that this is actually very similar to the way that we as human architects build strong structures that need to be light but also strong. We tend to use for this beams and cables, like in the example of a bridge or the Eiffel Tower. So actually our body uses a very similar principle. By using fibrous scaffolds, the body is able to make tissues and cells that are very strong, which is important because, for instance, your lungs breathe in and out, which stretches your cells continuously. In your blood vessels, there's fluid flow, and in your knees and in your spine, there's a large compression of your tissue. And somehow cells and tissues need to resist this, and they use the fibers to do that. So the question that we study in my lab is, what are the design principles of these networks? And this is in principle a very fundamental question. It was already mentioned before by Joris and Sten that nanobiology is really about fundamental science of how life works. But of course, it also has connections to many applications. And one of the applications we are very interested in, and I will come back to that at the end, is the origin of tissue diseases like cancer. And another application that you can think of is that if we understand how nature makes materials, we can maybe use the design principles also in synthetic materials. Now, what we want is to find somehow universal design rules. And that maybe on first sight seems very tricky, because if we look at our own body, there are 200 different cell types, at least 25,000 genes. It's clearly extremely complex. So yeah, what is the prospect of finding any universal design rule? But if we switch actually from this biological perspective to a physics perspective, we can actually find some universality. Because as I said, the cytoskeleton in the cell, the matrix, they're basically made of filaments. And in physics, we have ways to think about the mechanical properties of filaments, and this is called polymer physics. And we find it back all around us. Think about a rubber band, think about pudding. All of this is made of filaments. And that's actually not so different from our body. And using this kind of reductionist perspective is a very powerful way. Sorry. It's a very powerful way to find the design principles of biological matter. So the way that we approach this in practice is that we actually make minimal model systems. So we take a test tube, we purify the proteins that are in our body, and we reconstitute simplified networks that represent those networks that we have in the body. And the reason why we do this is obviously because this is now a very simple model system where we know the composition and can measure the properties. And this allows us to directly compare to theoretical and computational models. So we can actually get a very quantitative understanding of the mechanical design of life. Now, I mentioned before that one key design principle that nature uses is to use stiff beams and cables, a principle that we also find in human architecture. But if we look more closely, actually life is quite different. So a human architect would use very stiff beams and cables. And also, when you put these beams and cables together, an architect needs to use a very high connectivity, because otherwise you get structures that are very floppy. And when you look at nature, this is actually very different. The filaments that we find in the cell, which are nanometer sized, are actually very flexible, which you can see in this little movie where we have one of these filaments labeled with a fluorescent dye. And the reason why it's so flexible is because it's on the molecular scale. And another key difference from the way that a human architect builds is that the connectivity of these filaments is actually very low. So this is an electron microscopy picture of the cables that we find in the extracellular matrix. And you see that where the cables come together, these are the junctions. This is actually only 3-fold or 4-fold, which actually, if you would build that with Meccano, it would not be stable. And this is a very surprising thing. Nature seeks out the edge of mechanical stability, and that's what we found in our lab is actually key to making your tissue soft but also stretchy. And this is something you can see yourself if you take a net and you try to stretch it like this. You see that initially this is very easy, it's very soft, but when you stretch a lot, it becomes very rigid. And what you can appreciate when you do this little experiment is that it's soft initially because the cables can reorient, but when you stretch a lot, the fibers are all arranged along the stretch direction, and this makes it very resistant to stretch. So we found actually by combining experiments on these protein networks with simulations. So here you see an example of stiffness as a function of deformation. So initially stiffness is low and then it increases. And we find actually perfect agreement between experiments and computer simulations, which is quite surprising given that this is such a simple model. But there's of course even more tricks that nature has up its sleeve, because biopolymers in our body are also, they have a very complex molecular structure. and this gives them extra resilience. And this is something we can find experimentally when we use so-called optical tweezers. So optical tweezers are laser beams that we focus in a microscope, and we can use the laser beams to manipulate little micrometer-sized particles. And in this experiment, so this is a real lab experiment, you see here a little protein filament that is suspended between these two beads, and we are using light to stretch the filament. And you can see that it's actually very stretchy. We are able to stretch out this filament a lot, and we can also do it repeatedly. And again, we can compare this against computer simulations, which you see here in the bottom, where we can take into account the full atomic structure of these proteins and run a simulation experiment. So in the computer, stretch these protein filaments. And then we see that, again, these filaments are very stretchy, but now we can see why this is happening. This is happening because the molecular structures can actually change under the effect of a tensile force. So nature somehow has a very smart design of its materials. Now, of course, cells are not just materials. They are also very dynamic. And you can see here a microscope movie where you see cells taken from the body, put on a cover slip, and you can see that they spontaneously crawl around. And this is because the cytoskeleton that is inside them, this protein network, is not only forming a network, but it's actually also the motor of the cell. It's driving motion. And it is a motor, but again, it's a very different sort of motor from the sort of motor that we put in our cars. So in a car, we put a motor that is put together with one purpose. But in the cell, we have a protein structure that can actually switch between different purposes. So the cytoskeleton can, for instance, make a ring that divides the cell so that the cell can divide in two, but it can also form a machinery that the cell uses to move around. So this actually means that cells are able to reconfigure their motor. And this is because they use nanoscale components. So maybe you have heard of motor proteins, which are little proteins in cells that are able to use chemical fuel in order to move around in the cell. And using cytoskeletal filaments together with motors, the cell is actually able to self-assemble into different structures. And this is something we can, again, in the lab reconstitute. So we can take the motors, we can take the filaments, put them together, and here in the microscope, you see that this spontaneously organizes into a structure that can contract like a muscle. And again, this is something we can also do in a computer. We can take, again, motors and filaments, and we see in the computer that this spontaneously contracts like muscle does. So yeah, this is illustrated with this kind of approach. You can actually learn a lot about how biology works. But of course, we are still quite far away from this big question, which is how lifeless molecules are not alive, can form a cell that somehow is alive. So how do we transition from the domain of chemistry and physics of molecules to the domain of biology, where we have a living cell? This is something that we are very, very intrigued by. And together with us, many other groups in the Bionanoscience department in Delft are interested in this question, and also groups elsewhere in the Netherlands. So we have formed a large group of researchers that work together on this question. And what we are trying to do is figure out if we can actually take cellular components and build a synthetic cell that is able to grow and divide. And you can think about this, as a kind of roadmap. We now know how to make cytoskeletal networks that are active. We are trying to encapsulate this into lipid membranes so that it starts to look more like a cell. And we try to integrate that with other modules, such as metabolism and DNA replication. And then hopefully, in many years probably, we can get closer to this question, how cells have their magical ability to grow, divide, and self-renew. So this is one direction for the future of nanobiology research. But of course, there's also directions that are more closely related to biomedicine. So we are also interested in the question if we can use fundamental nanobiology and biophysics to improve human health. And one area where I think we can make a difference is in the area of using stem cells for tissue repair. So stem cells are cells that are able to differentiate into all the different cell types in your body. And traditionally, people think that they need some biochemical factors, some growth factors to induce stem cells to differentiate. But more recently, it has become clear that stem cells can also be triggered by mechanical cues. So if you take cells and put them on a substrate, on a gel that has a different stiffness, similar to brain or fat or bone, you can actually trigger the stem cells to become more like a brain cell, a fat cell, a muscle, or a bone cell. So you can actually use the natural stiffness that we find in different tissues and use that to program cells. And we want to figure out how that actually works, why is this happening, but also whether we can maybe use that. So can we use it together with researchers at the Erasmus Medical School to make artificial tissues that we can use to repair damaged or aged tissues, or that we can use in personalized medicine to test drugs. And another direction here is that mechanics are also very important to make the difference between a healthy cell and a cancer cell. So we are trying to figure out what are the mechanical origins of, in this case, glioblastoma, which is a devastating cancer in the brain. So I hope I've, in this case, made the subject of nanobiology research a little bit more alive to you. And what really draws me as a scientist to this multidisciplinary field is that it's extremely cool to learn and somehow put together ideas and concepts and methods from physics, biology, mathematics, chemistry, engineering, and medicine. And the people that actually work in my lab come from all these different disciplines. We talk to people from all these disciplines. And the exciting thing is that this sort of allows you to constantly learn new things. So I would like to leave it here, leave you again with this page so that you can look up more details. And I'm very happy to take any questions that you may have. Are there questions in the chat?
Yes, there are a couple of questions, however, not specifically about your presentation as far as I can see for now. So from now until 10:30, we will be able to answer questions, both about this presentation from Gijsje, but also about a program or about a presentation from Joris or from me. So we will now go over the questions that are asked. Feel free to answer feel free to ask any more questions. And again, in about 5 minutes, I will send my email address for any unanswered questions or any questions that you might have in the future. You can very informally send me an email and I will reply to you for sure. So one of the question now is whether there are any students who have already received a bachelor in another unrelated subject or looking for a career change. Joris or Joanna, would any one of you care to answer this question? I see Joanna would like to say something.
I was gonna let Joris do it, but if he's doesn't have good answers either. I'm not sure I have good answers. I know people look at doing it. I think there are students who have done multiple bachelor degrees. Oh, Joris has got an answer.
You're muted, Joris.
Okay. Okay, again. There's only one example I know of most students we have, as was already said before, are directly from high school, so 18, 19 years old. There's one person, one former student I know of. She did biology as an HBO study, so a bachelor. Then worked at a patent lawyer office for a while, and then she saw that there was more and more patent applications in biophysics, nanobiology-like type of directions, and then she decided to start the nanobiology program to really train herself in the concepts of nanobiology, so she could much better perform in her patent lawyer office for all these patent applications. So then she really understood where she was talking about. So yes, we have at least one case where somebody, well, has another career, but is now using nanobiology to help her in her current profession.
Well, that's actually very interesting. I never heard of that. That's very cool. Okay, these questions are already answered. This one too. How is your relation with other worldwide faculties, and is it possible to do your master's after this at another country/faculty? Yes, it is. Maybe someone else can say something about the relation between worldwide faculties, as I'm not entirely sure as to what you're referring to. But it is possible for sure to do masters internationally or in the Netherlands at other universities.
We have several connections to other faculties. So, for example, to do a minor abroad in Sweden or in some other countries. We often get questions from students whether they can do an internship abroad, so at another university, and all the faculty in TU Delft at Bionanoscience or in the Erasmus, they have a very broad network. So often students come talk and say, oh, I would like to do something like a project abroad in this topic, and then, well, there's always one teacher who knows somebody in another university that works on that topic and sends them that way. For masters in another country, there are clear intake rules for their application. So, and because this is an international master, you're eligible for several other masters in Europe and even outside Europe. So, but it all depends on what that master program is asking for background, knowledge, and courses. And those have to match with nanobiology.
Okay, next question is the following. If you haven't learned English yet, are there any courses to learn it, or do you need to study it by yourself? Assuming that if you have a high school degree, you have already had some English, which should be on the correct level. I see that Joanna has some additional information on this.
Am I live? No. Yeah. So you have to have an English qualification before you join the program. So that could be based on that it's your native language, that you did this in Dutch high school. If you're an international student coming in from not an English-speaking country, you will need to have passed an English proficiency certification to be admitted.
Okay, thank you, Joanna. Do a lot of students do double bachelors, including nanobiology? I'm considering doing that, but I'm not sure. Does anyone know this? I see that Joris does. Great.
We have a few students that do double bachelors. For example, nanobiology and applied physics. We had one bachelor that is doing actually a triple bachelor between nanobiology, applied physics, and math. So Yes, it's possible, but you have to talk to the programs first to make sure that it all fits in your program and in your week, right? So, but it is possible.
Great, thank you, Joris. Next question is, what do you do during the bridge courses of the program? So, like Joris already briefly explained, the bridge courses are courses where multiple disciplines come together. So, one course would be biomedical programming, for example, at the end of the first year, which is a programming course where you learn to program a specific, you learn to program specific biological situations. So this is an example where multiple disciplines come together, come to one. So we have plain math courses, plain biology, and plain physics courses, where the bridge courses actually fall together, if you will. Next question, what if you're bad at maths but very good at the rest? Well, what happened, I wouldn't say what happens a lot, but what happens sometimes is that people underestimate the amount of mathematics in the program. In the first year, there's a lot of mathematics. From the top of my head, it's 16 credits out of the 60 in the first year that is plain math. So that's more than a quarter, right? That's more than 25% of the first year. So given that there's a lot of mathematics involved, you might want to, give this some additional thought. And eventually, I mean, the mathematics works back to the rest. So it's an interdisciplinary program, and besides, the mathematics builds up to the physics, right? In the second year, you have a lot more physics rather than math, which makes sense, because in order to get the physics, you need some mathematical knowledge, some mathematical background. How small scale is the study? As in, will it be looking at only cells and their components and materials? Okay, this is a very broad question. How small scale is the study? Well, I would like to think that the cell is a central unit of the program, but it's very fundamental in a way that we want to describe the processes that involve cells and that involve the small scale biology, if you will. And so what we study is not just applicable to cells, but also a little bit to a higher and lower organization as well, as we learn a bit about quantum mechanics and about atoms. So it's a very fundamental biology. So in that sense, it is a very small scale. Does anyone want to add something? Joris would like to. Great.
Yeah, so Nano already says it, right? It is, let's say we mostly start at a cell with cell biology and then go smaller, smaller, smaller. We don't have that much physiology or insight, although we have development. So how do we develop and evolution? So these biological principles are also covered extensively in nanobiology courses.
Great, thank you. Here's a question for Gijsje. I can see that Gijsje is already typing, but I think it's still fun if she elaborates on this orally.
Yeah, so I received an interesting question whether this principle can be applied to repair some genetic defects in cells in the future. And I would say hopefully yes. So we are trying to identify the molecular basis of disease. And a very concrete example is, for instance, cancer cells, which misbehave because they metastasize. So they start to invade the surrounding tissue. And we try to figure out how the cytoskeleton actually drives invasion. And if we know which molecules are the culprits, then of course that gives you some therapeutic targets. And there's different ways that you could therapeutically target this, but indeed one way would be genome editing. And of course, also Sten, I think, mentioned it, CRISPR-Cas technology is, for instance, one of those technologies that are now advancing very rapidly. So I think there's good hope that knowing the molecular targets, we can then also use genome editing to repair some diseases at the core and not just fight the symptoms of the disease. I also received a second question. Well, maybe it's not for me actually. So I talked about stem cells, and the question is how much is taught on basic cell structures in the first year? So this is maybe a question I can give to either Sten or Joris. Joris.
Well, in the first year, but also in the second year.
Get live, Joris.
Oh, in the first year and in the second year, your biology courses really are aimed in improving your overall understanding of biology. So it is about genetics. How does DNA work? How is information encoded there? How is it read by the cell? How do proteins work? Cell structures, how cell division works. So in these courses, you get a complete overview of the basic principles of biology at a much deeper understanding than your current high school level. And based on that, you can do very specialized courses again. Anything I forget, Sten?
No, I think you pretty much covered it. So there was a question a while back which hasn't been answered yet, which is how many students apply for the numerous fixes. Maybe Joris can say something about this.
Yeah, so this year we had 209 students that finished the numerous fixes selection. So that is both the questionnaire and the selection exam for 120 first year spots. So then you know a little bit the ratio. We know from experience that if your number, for example, this year we had only 100 spots available, so we will increase. And up till rank number 153 have been awarded, because not everybody who gets a high rank or low rank, so you're eligible for nanobiology. Some people decide not to do that or do another study or some, so they drop their rank, and then the next one on the list will be invited to apply for the first year. So this is a little bit, as I said, this year up till 153, rank 153 was included in the first year, and then this year we only had 100 students. So then you know a little bit, yeah, how much chance you have to be a first year student.
Thank you. Okay, so it's 33 now, which means that we will still run along for about 10 minutes. So for any questions still, please do share. Yeah, and if you need to leave, of course, that's fine. Next question is whether it's helpful to have Math D alongside Math B. So I did it myself in high school. So for the ones that don't know, Math D is just like an extra course, an additional course in math, which covers a broad range of things like imaginary mathematics and stuff. So yes, it is helpful. Necessary, definitely no. Everything that you need to know for Math D is covered in the first mathematics course, Analysis 1. So it does help because you have already seen some abstract mathematical ideas, and so you might already be used to some bits, which makes rolling into the university mathematics might be a little bit easier. But you would be perfectly fine without Mathematics D. Yeah, we take you by the hand in that. You are taken by the hand in that matter. So without having done Math D, you don't miss much. Next question. If I am not taking biology, my last 2 courses of high school that I did before, can I still apply? This is very specific. Joanna or Joris, does any one of you know? Joanna?
you have to have biology. I can find the exact requirements here because I was just looking at it. Yeah, so you have to have done biology in high school. I'm not sure I can find the exact level just off the top of my head, but I'm going to post a link in the chat here that's got what you need for a Dutch diploma.
Okay, great, thank you. I just made an announcement with my email address. I had hoped to do it earlier, but here you go after all. So for any unanswered questions or any little wonderings or interesting questions or interesting ideas or whatever, you can informally contact me via this email address. Yeah, so feel free to do so. I will definitely get back to you. We don't have any open questions. So if there still are some wonderings in the last few minutes, don't be afraid to ask. We are here to help you guys, so please do share. If I'm not taking physics as my last 2 courses of high school, can I still apply? This is the same question that we just had, but then biology is interchanged with physics. And the same goes for physics. So you do need to have physics in order to be eligible for the program.
So there are no more questions. I would like to thank you for being present here at this Open Day Nanobiology. I hope you have now a much better understanding of what nanobiology is, what we study, what it needs to do nanobiology. And of course, we all hope to see you in the future live in classes and being one of our students. So thank you for your presence and have a good day.